Okay, let's resume chapter 14. I guess this is the second part of our discussion. And we had just gotten through, if I remember right, yeah, we had just gotten through talking about isoelectric points and how, how that influences the charge state of a protein, and of course an amino acid before that. And so we're meant to then continue by just, again, we build in complexity here. Again, back to carbohydrates. I'm thinking of where we started out with a monomer and then they got put together right into these larger and larger uh, uh, polysaccharides. Same thing here. Now we're talking about putting amino acids together, and we just did that. I think we drew a tripeptide in our last example, put one of those together. Now we're talking about a little bit longer strings of amino acids. And what we encounter is these different levels of structure as these things, by that I mean peptides and proteins, get larger and larger. Remember, the distinction is 50 amino acids. It's, anything greater than 50, we're calling it a protein. 0 to 50 amino acids, we're calling a peptide. So so we're going to go through each of these individually. I don't know that I'm not going to read them to you, but when we're talking about primary structure, we're literally talking about the sequence of amino acids. Remember, there are 20 of them, naturally occurring ones. And so one of the things that we'll kind of see, in fact, it's on one of these upcoming slides, is there's so much complexity that can be derived from 20 different amino acids put together in so many different ways, and that's a really important thing for us to discuss, and that's really the subtopic of primary structure. Secondary structure, there are a few types that I want you to know: alpha helices, beta sheets, and random coils, and they're listed here, and again we'll go through each of those in detail. Tertiary structure, the overall three-dimensional arrangement of a single polypeptide chain. And then quaternary structure, if you have two proteins or peptides, two polypeptide chains or more that come together and kind of aggregate, and there are different forces that hold these together that we'll discuss, then you have quaternary structure. Okay, so we'll start with primary structure, and here we are. And again, there's this, here, I'm going to get the laser pointer out here. There's this mathematical expression, really, but what it's getting at is, you can read it there. I mean, the statement is there. If you have a certain number of amino acids, call that X or call that N, 20 to the N gives you the number of possible peptides of that length, different ones, right? And so as these things get bigger and bigger and bigger, you can imagine all sorts. And here's that kind of a specific example, kind of spelled out for us. A huge number of tripeptides, and even huge number of tetrapeptides or pentapeptides, right? And in many cases, if we're talking about even proteins that are 150, 200 amino acids long, and that's not even huge necessarily, our body can produce one of them via these metabolic pathways. And so really amazing stuff here. Okay. Oh, wow. Yeah, here is another incredible kind of statistic there. So 20 to the N, really interesting application of primary structure here. Now, what do I want you to know? Well, I want you to kind of understand, right, that there's so much molecular complexity here involved, even when we're talking about primary structure as it relates to the different kinds of proteins that we can make. But another thing that I want you to be able to do, and if you take a look down here at this figure, these figures here, is I would want you to be able to take, an amino acid sequence, a polypeptide chain that's 3 or 4 or 5 or 10 amino acids long. This one, it looks like it's 16, and I would want you to be able to give me its sequence. Okay, now, how do we do that? Now, hopefully we'll remember, right? We remember how these things are put together, and we remember that it's really the R groups that distinguish one amino acid from another. And also, let's remember that when we write these things out, we write them from left to right, right? N terminus on the left, C terminus on the right. So, and I guess I can kind of, so if we're looking at this specific example, right, there's the N terminus, or the nitrogen, the amine group, right? That's part of the N terminus, and here is the C terminus, right? The carboxyl group. Okay, one of the things that I want us to be able to do is, if we're given, on an exam or on in an exercise, is if we're given a sequence of amino acids, 3, 4, 5, 10, 15 amino acids. In this case, it looks like this one's 16 amino acids long. I would want us to be able to give or identify or write down the amino acid sequence, okay? So, and again, that's primary structure. It's which amino acids are present, are linked together by peptide bonds. And we do it, remember, from the left to right, N terminus to C terminus. So in this case, what I want to do, and remember, it's R groups. So I'm going to circle R groups. I won't do it for all of them, maybe just the first 3 or 4 of them. Okay. So there they are. We're going from left to right, M terminus to C terminus. There's another one there. That's the same as the first one, it looks like. And then we'll just stop with this one, okay? Now, again, I don't expect you to have these amino acids memorized, but just CH2OH, okay? It is serine, but I want to just make sure you know where I'm getting that from, right? So let's go back to these amino acids. And again, you don't have to memorize these. These are going to be made available to you on an exam. So here we are. Let's look for serine. Okay, so you see how it's the CH2OH, right? That distinguishes serine from any other of the 20 amino acids. And so that's really what I'm looking at here. That was the first amino acid. Sorry, I'm scrolling all the way back through this. Where are we at? Here we are, right? That's CH2OH, there's serine, right? And if you're looking over here, right, that's the first amino acids that's listed. Now, you could list these by name, right? Remember, amino acids have, they all have a distinct name, then they have a 3-letter abbreviation and a 1-letter abbreviation. So I want you to be proficient at all of those, but again, you'll have all of those in front of you. In this case, it's just the 3-letter abbreviation, and they have these weird brackets on them here. I think this example I took from online. I was looking for one of these, and by the way, we've seen adrenocorticotropic hormone before, and so I think that's why I chose this one, is because in a previous chapter we had talked about it. It's a peptide hormone, right? Remember, hormones are signaling molecules, and this one happens to be a peptide, so it fits nicely here. But anyway, they don't always have these weird squiggly brackets. Sometimes there's just a dash between the 3 or the 1-letter abbreviations, or the full name if you want to use the full name. Now, this is the next R group. So again, if you're going to your list of amino acids, you're looking for a CH2 attached to a benzene ring attached to an OH, and of course that would give us tyrosine. Or Y. Y is a 1-letter abbreviation if we were using those. Now, back to the next one, it was that CH2OH again, it's repeated. So notice there's serine again, okay? And then if you look at the CH2, CH2S, CH3, if you're going to amino acid list, you'll realize that one belongs to methionine, and we go on and on and on, okay? So that's something that I definitely expect you to be able to do. If I give you an amino acid sequence, I want you to be able to decode it, or give me the primary structure, the sequence of amino acids. Okay? All right, so let's see what we got next. Okay, so yeah, this is just an example. It's a figure from our textbook, and it's meant to illustrate that even small changes in amino acid sequence can lead to structural differences that lead to even functional, I should say, differences. And what you see is these are different forms of insulin. The reason this is a good example is because a while back, this is probably, I don't remember, I think it was like early 2000s. Before that, it was acceptable, and it still is, I think, in some places in Europe, to use insulin derived from other animals, right? In this case, you see it looks like humans and cows and pigs and sheep. Those are the ones that we're looking at here. But in the U.S., you have to use. So this is believe it or not that hard. It's not that hard to make insulin in a lab. So we use, if we need to supplement our insulin, we would use synthetic insulin, stuff that's made in the lab, and it's human insulin, it's just a synthetic form, or it's synthesized in the lab. But it used to be that there were these actually cocktails of insulin that would act differently, because some of these, human pigs, sheep, cows, the insulin actually in our bodies actually acts a little different in terms of how quickly it does what it does, which is right, it helps sugars get into the cell to be metabolized and all the rest. But really what we're focused on here is, okay, there's subtle differences in amino acid sequence, and also not all, this is kind of a weird one, because you have two polypeptide chains that are held together by disulfide bridges. We'll talk about disulfide bridges more in a little bit. But a lot of times if you have a peptide, it's just a single chain. This one happens to be two of them, an A chain and a B chain. And the question is just, okay, what form of insulin is pictured? And this is, again, just in exercises. Can I kind of look through this sequence of amino acids and note that there are differences? And those are, of course, primary structure differences. So I'm looking at them, and to me, I'm looking and I'm saying, okay, let's look at, it looks like for the A chain, we're looking at amino acids 8, 9, and 10, going from N to C, right? 5 and then 6, 7, 8, 9, and 10, alanine, serine, valine, it looks like. So right away we're like, well, this must be cow, right? But let's confirm that. If we look at the B chain and look at, amino acid 30, it is definitely an alanine. So there's 5 and 6, 7, 8, 9, and 10, alanine, serine, valine on the A chain. And of course that corresponds to the bovine form. Okay. All right, what's next? All right, so we'll move on to secondary structure. Now, secondary structure, secondary, tertiary, quaternary, we're talking about three-dimensional regions of space, but, or I should say the overall conformation of these big polypeptides, but secondary structure refers to these very specific regions. So there are these different types of structures that repeat themselves in lots of different proteins. The three that we'll focus on are the alpha helix, the beta sheet or the beta pleated sheet, and then random coil. And I know random coil sounds like, it's weird because it's like, wait, that's like, it's randomized. Why would we call that a repeating or a uniform type of secondary structure? And some people wouldn't. Some people would say random coil isn't really because it can look different in different places, it's not really recognizable. But we're gonna include random coil in our discussion about secondary structure. So we'll consider it a third type of secondary structure. Now, again, we'll go through all of these, and you see descriptions of alpha helices and beta sheets. And maybe let's just start by, we've got some nice figures here we can refer to. Okay, so an alpha helix, and I'm gonna try to draw one out. As well. It's not always a beautiful thing, but there are some things that I want you to know about alpha helices. And so there's this, it's a helix. It propagates from, well, I always think about it from top to bottom. It's kind of a coil that works its way down. But you see an example of that here, and it's held together by hydrogen bonding. So it's like a right-handed helix or spiral. Okay. And again, we've got more. Let's go back here to the pen. Got more examples of this. Okay, now there are some pieces of information, right? This is biochemistry, so there's a little more, biology, functionality, memorization involved. I do want you to know these. There's just a small subset of kind of factoids about alpha helices, and I want you to know them, and they're listed here on the slide. Again, I'll refer back to these, but I'm gonna try to draw my own alpha helix in a minute. And here's another nice depiction of the alpha helix. So I think I have I have a blank sheet here. So it's not going to be beautiful because you all know that I'm not a great artist, but I'm gonna do my best here. And I want to point out a few things, primarily how these amino acids are. Some of them are held together by hydrogen bonding, and that really holds the whole helix together. So let's do this: anatomy of an alpha helix. Okay, and so again, if we're thinking about this as like this right-handed spiral or helix that kind of propagates, you know, from one direction to the other, we would just think about it coiling down, right, from the top to the bottom. Or let me just get rid of that little. That's weird. There it is. Okay, I was like, where did my eraser go? Okay, so or if you want to think, if you know, if you're looking at like that, this is like side view, I guess. This is like side view. Okay, if we're looking at like top view, it's this, right? And if we're just taking like a chunk, and here's what I really want to try to do is take like a piece of this and expand it. Maybe I should have made myself a little bit more room here. Let's do top view a little bit smaller. And you get the idea. And then let's take this and let's try to expand it. And here I'm gonna give this a go. Okay, give me a second here, because I'm even worse if I try to talk. And okay, there's a peptide bond, right? And I'm gonna make these R groups generic. So here's another thing that I'm trying to point out as I draw this is that the R groups are kind of around the periphery of the spiral. They point outwards, and the NHs point up, and the C double bond O's, the carbonyl groups, point down. And those are the things that hydrogen bond. Now, not every one of them hydrogen bonds, but some of them do. Okay, so again, here we go. So carbonyl, another carbonyl, right? Next thing, that points down. Next thing would be a nitrogen, hydrogen. That would be you know, pointing up, as it were. And then again, we're making these. Notice how we're making the R groups generic. That's okay. Whoops. Just make that a little. Okay. The next thing would be a carbonyl, another one, right? Pointing sort of down the way I drew it. And then next thing, nitrogen, I guess hydrogen. And then, sorry. Again, my coil isn't beautiful here by any stretch. So let me try to draw this R group now pointed this way. So we'll use kind of a dash to do that. And then I guess next thing would be we're back to a carbonyl, right? Pointed down. We're still kind of working our way this direction. That's not too bad. And H pointed up. And then it would be another carbonyl, right? Did I get the R group in there? I'm trying to think here. Yeah, okay, that's okay. And then back to the R group. And then we'll just pretend, right? We'll pretend this. Oh, I'll just draw it as a, so, all right, we'll pretend this continues. Now, couple things, and same thing here, I'm just pretending that this continued here, right, and continues here. Now, couple things that I want you to know. Now, this is not drawn perfectly to scale, for sure, and if you look back at that slide, I said I want you to know those details, approximately a 3.6 amino acids per turn, and all those sorts of things. And this is the thing that I want to add to this picture. So I want to get the red here. I want to show you that there's basically a carbonyl and an NH hydrogen bond. And a carbonyl hydrogen bonds with an NH that's 4 amino acids away. So if we're kind of circling these amino acids, I guess it would be 1, 2, 3, and this is the 4th one. And so it works out kind of nice that it's right underneath, and that's exactly what's going on. That's exactly how these things are held together. So right there, right, that's that H bond. And there are many, many of these that help keep this helix constrained, okay? Meaning keep this polypeptide chain in an alpha helix. Okay, so again, not a perfect representation, but not too bad. I've done worse, that's for sure. So anyway, here's again back to those details. Okay. Okay, so alpha helix. All right, next thing, right? We said 3 of them: alpha helix, beta sheet or beta pleated sheet, and random coil. Okay, so all right, so a few things about the beta sheet. A lot of times they are, you know, you see these cartoon representations because these are huge molecules. We could never hope to draw them out, right? I mean, look at how I just struggled with a little 4 amino acid section of a protein. So a lot of times you do see these cartoony figures. And we'll do some of that as well as we try to distinguish between what we call parallel and anti-parallel beta sheets. But yeah, you see, again, it's hydrogen bonding. And I know that there's a little arrow there. I think I have a better one. Yeah, here's a better one where you actually see the little red dashes that indicate the H bonds in these different versions of beta sheet or beta pleated sheet. And what you see here too is just a few other little factoids that I want you to know them about beta sheets. Again, it's the NH and the C double bond O of the different the peptide bonds, right? Because those functional groups are part of peptide bonds. Okay. So again, I want to try to, and I have another blank sheet here. I want to do the same thing. It won't be quite as sophisticated. It'll be a little bit more cartoony, but I do want to show you, make sure we're clear about the difference, especially between parallel and anti-parallel beta pleated sheets. So let's do anatomy. Whoops. Anatomy of a beta sheet. Oh, or again, beta pleated sheet, same thing. And we'll just put parallel, parallel versus anti-parallel. Okay. Okay, so let's try this. All right, here we are. Now, the main difference here, and by the way, these are just big chunky arrows that are meant to represent the way this thing is pointed, starting with, and I want to make this clear, because you saw a similar thing on the last slide, but to me it always helps when I'm thinking about these. I'm writing them, remember, N to C terminus, left to right. And in this case, what the distinguishing feature here is that the N and the C in anti-parallel beta pleated sheets, so I'll write, I should have, maybe, I could have given myself the anti-parallel version, I could have given myself a little extra room here. The N's line up with the C's, so this is random coil, by the way, our first example of that. And again, if we're heading in this direction, left to right, as we propagate down, we're continuing here. Here's some more random coil. And again, it would continue on. And same thing here, continue on. But one thing you notice, again, is that the N's and the C's well, the N's line up with the C's and the C's line up with the N's. Now, let's think again about how these are held together. Remember, it's those carbonyls, and we're not going to draw them here, but it's this, right? It's those hydrogen bonds that are holding these things close to one another as they line up, okay, in an anti-parallel way, okay? So if we jump over to parallel, right, you'll see that it's very similar, but the distinguishing feature, and this is why I write it out, I always like to show you the N's and the C's, because that's what helps me recognize the difference, is here, right, if we're thinking about our and if you actually do write the arrows, like we're doing, that's a helpful distinguishing feature as well, because the arrows are going in the same direction as as compared to in a parallel version, as compared to the anti-parallel, which we just drew. Notice that the arrows are pointed in opposite directions. So here the random coil has to loop all the way around. and there's another N and another C. Notice how the N's and the C's are lining up, or notice how the arrows are pointed in the same direction. So same thing back to here. Oops. N and C. And again, we could just continue that. We'll just stop it there or stop it here. Notice this then is the parallel version of a beta sheet. Beta sheet. This is parallel beta sheet. Okay. And we can just write, these here are, oh yeah, let's not forget, these are the hydrogen bonds. Now, I know the random coil seems like it's in the way here. It certainly wouldn't be in the way. You could imagine that being above or below, where these hydrogen bonds are forming. So that's not, it looks like it's getting in the way, but it's not. And we can also say too that, so obviously these are H bonds. So we've discussed all these red dots, and we can also write just regions of random coil. Coli. Just to kind of point those out as well. Okay. All right, so beta sheets. And this, I think, is just a figure from, I think it's probably from Bettelheim, from our textbook. But you notice all three. And again, they're often depicted in this way. The coils are really, these curls that are kind of easy to identify. Remember, this really represents a big molecule, a big long chain of amino acids. But again, it would be really hard to recognize anything meaningful if we just had all the amino acids, right? There could be hundreds of them in this protein. We could look up carboxypeptidase, and I'm betting there are. I'm betting there are, anywhere between 50 and way more than that probably. Anywhere between 100 and 200, maybe more amino acids if you look up carboxypeptidase. I don't actually know offhand, but what you can see in this figure is, right? Notice how you see those kind of flat regions, or the arrows. Those are, of course, the beta sheet. And you see the coils. Those are the alpha helix. And then you see the random coil, the orange regions in this case as well. So anyway, just an example of that. Again, one of these cartoony protein figures. You see that red dot in there as well. That's probably a coenzyme or a cofactor. It might be a metal ion in this case. And we'll talk more about that here in just a little bit, and in our enzymes chapter, because carboxypeptidase is actually an enzyme, but all enzymes are proteins. So again, more to come on that. Now, we're at tertiary structure. So again, the overall three-dimensional arrangement of the polypeptide chain, whether that actually be a peptide, a larger peptide, or a protein, it's one polypeptide chain, and that's where this is distinguishable from quaternary structure, as we'll see. But let's say this before we look at this. Is just another figure from our textbook that we're meant to kind of decode. But let me do this real quick. Let's do a little bit of work. And let's say I want to talk about the actual fundamental forces or types of interactions that hold tertiary structure, that overall three-dimensional conformation of a polypeptide chain together. There are really 5 fundamental types of intermolecular forces. I almost think, if I remember right, it's like in the back of my mind that Bettelheim doesn't include disulfide bridges. He may or may not, I don't know, but I want you to think, okay, there are 5 types of forces. These are in what we call intramolecular forces. In fact, let me just go ahead and write it, and we'll talk. So 5. Okay, so tertiary structure. Okay, and let's put 5 types of and notice how. Okay, so I'm just going to put forces or interactions. And I was going to actually write intramolecular forces, but this becomes a little weird just with the verbiage. Technically, remember, inter is between. We've talked about intermolecular forces our whole, the whole course, right? And so we're going to see that some of the, like hydrogen bonding is one of these, and London dispersion is one of these. And so if I called them intramolecular forces, you're like, wait a minute, we've been calling those intermolecular forces the whole time. Now the force is the same. It's just that when we would call it, if in this case we called it an intramolecular force, all we mean is a force between, or excuse me, within a molecule. And if you think about it, even though we have a whole bunch of amino acids that are all linked together into one big molecule, right? These polypeptide chains are technically one big molecule. So really, technically, these are intramolecular forces, but let's not confuse the issue. All right, so 5 types of forces or interactions. We'll say that stabilize the tertiary overall 3D structure. So getting kind of sloppy of a protein. Okay, and let me just say this before we list them. These interactions, forces, whatever. These interactions. Whoops. They're mainly occurring between the R groups of amino acids that are close to one another. That's really where, these stabilizations occur, stabilizing influences occur. It's not that you couldn't include the carbonyl or the NH, but generally speaking, what we're talking about now are R group interactions. So let's say these interactions are generally occurring between R groups of amino acids that I guess we could have put adjacent amino acids, but let's just put that are close to one another. Okay. Now you'll see where we're going with this. We're going to need to do some practice. Some practice anyway. But here they are. I want to just list them for you. Okay, so they're whoops, and we'll go back and practice because you see some of them here, or we would see some of them here. We should be able to identify those. But let me just list them first. So one that we're very familiar with, H bonding. That's one of them. Now, sometimes it's weird in biochemistry. Sometimes we use different verbiage to describe things we've already learned. So I think typically Bettelheim and in the biochemistry world, salt bridges are often. So I'm going to write that. Salt bridges are one of these. But we know salt bridges. We know that a salt is just an ionic compound, and that's really what these are. These are just ionic bonds. So AKA ionic bonds, AKA electrostatic attractions. Yeah, there's a lot of ways. Sorry. So these are described. So anyway, if you see any of those, they all mean the same thing. These are just ionic bonds. Okay. Another one that's kind of like that. Whoops. Again, it's just kind of silly and sometimes annoying. But another one that you see, it's just these words hydrophobic and hydrophilic are so prevalent in biochemistry, and for good reason. But sometimes again it's a little annoying. So hydrophobic interactions. Hydrophobic means, remember, nonpolar essentially, right? And so if we think about well, let me write it. If we think about interactions between nonpolar things, right? What we've been conditioned to call those forces or interactions is London dispersion forces. So again, AKA London dispersion, or just dispersion. Spell it right. London dispersion forces, right? So again, those mean the same thing. Now, I don't think we've seen this one, metal ion coordination. This would be another one of those 5. So these are also, well, okay. So metal ion coordination, again, is what I think what Bettelheim calls them. You can check me on that. Also called coordinate covalent bonds. That's what I would tend to call them. But here's the deal with these. We have not, even in Chem 3A, we don't talk about these types of interactions because they're really, there's a distinction that are made. They're really just covalent bonds, and we know what those are, but they're kind of weird in that they involve a metal, and one of the chemical species that's attached to the metal brings both of the electrons that form the bond, and the metal brings none, and they form this new bond called a coordinate covalent bond. But I don't care that you know any of that. I would probably encourage you to call them metal ion coordination interactions, because that's going to remind us that a metal is involved here. And that's really the thing that should jump out to us, because we can spot metals, and if you can spot a metal in one of these interactions right away, you're like, okay, metal ion coordination. So let's call them metal ion coordination more than anything, because that'll jog our memory. Okay. Now the next one is disulfide bonds, or disulfide bridges, sometimes they're called. And these, if you'll remember, are they're a very specific one. They occur between two cysteine amino acids, and the bond looks like this. I know we've encountered this. Well, I'll just put R here. Really, these end up being CH2s, but that's the disulfide right there, the two sulfurs, right? Disulf is where this comes from. But that's the idea, and we'll again be able to. So here's another one, right? It's very distinct. It's like it always looks the same. It's two sulfurs bonded together, and those sulfurs are bonded to two carbons, and so you can't miss this one, especially if you know the name kind of gives it away as well. So. And that's the one, I think, if I remember right, in some cases, textbooks, certain places that you look, it won't be listed as one of these stabilizing influences for tertiary structure, but it is. It's generally accepted as one of those stabilizing influences, and we're going to include that in our list. So if we go back, right, we're looking at these, and I'm going to grab the highlighter here, and what we're trying to do now, again. What you see here is pictures of R groups. And remember, those R groups are attached to amino acids, but it's really the R groups that are interacting. So we're kind of testing ourselves, and we're saying, okay, what are these interactions that are holding these amino acids, or the R groups, right, of the two amino acids that are in close proximity? What are they? What stabilizing influence of the ones we listed is holding these two amino acids together? So if you look at this is, and you can go identify these. In this case, it's not necessary, a serine and a tyrosine. You can go look at the amino acids and identify them. But really what we're interested in is that interaction right there, right? And so I'll get a red. And I'm going to say, okay, what is that one? And I think, again, probably what I'd encourage you to do right now is pause the video and think before I tell you the answers here, or we talk about them. Maybe just try to guess at them, because again, this is one of those things that I want you to be able to do. Maybe pause it and see what you come up with on your own, and then continue. But this is spoiler, this is an H bond. Okay. Now, if we go to the next one, right, the interaction that we're looking at is right here. And one of the things that gives this away is that this is an interaction between, notice, charged, positively and negatively charged regions of amino acids. Okay, and so in this case, I'm hoping that you'll agree with me and say, well, that's an ionic bond. It's a plus and a minus, right? It's a positively charged thing and a negatively charged thing. And let's call it a salt bridge. But again, we know it's an ionic bond or an electrostatic attraction, okay? Now, this one you don't actually see an interaction, and we want to actually write that there is one, but it's not, it'd be nice if they actually, had put a dotted line there too, and we'll just add it ourselves. because there is an actual interaction. Now this is the weakest of those, right? And the thing that you have to see here, recognize in order to remember this one, is that if you look at these groups, right, and this goes back to like, you know, our first, well, all the way back to 3A really, but especially our first chapters on alkanes, all you see here in these R groups is carbon and hydrogen. So we notice, right, that they are nonpolar R groups. And again, an interaction between two nonpolar things can only be one type, London dispersion, or in this case, again, we'll call it hydrophobic. Don't be surprised if, again, you see London dispersion, but hydrophobic interaction. Okay, now how about this fourth one? And I was going to say, well, okay, so let's just do this one, a couple of other comments afterwards. But what do you think here? Okay, and again, maybe you've done this already. Right away, you see that metal ion, right? Magnesium, you look at it, and you have your periodic table. So if you're ever sure you see this atom or this ion, and you're like, well, crap, is that a metal or a non-metal? You go back to your periodic table and you figure it out, right? Because we know where the metals are and the non-metals, and magnesium certainly is a metal. And so this can be none other than, right? And there's really two regions here. They're both part of the whole thing. Maybe I'll just circle the whole thing. Maybe that's better. And we'll just call this metal ion coordination. Okay, now, one of the things, and I don't know, okay, yeah, here's another example. Again, just, and these are all listed for us. They're all actually all identified. I'm just looking at them. I think this is a figure from, again, our textbook or some other textbook. But notice how, let me get the laser pointer here. And they're all listed, right? Here's a metal ion coordination. Leucine and valine and isoleucine, if you look them up, they're very nonpolar. So, right, that's really what we're seeing here. Now, again, there are no dots. It'd be nice if they added some, like a different color dot, just to indicate that there is an actual interaction there. It's a weak one. It's weaker than the others, but it's there. There's a disulfide bond, okay? So notice that, right? That's that SS, and each of the S's are covalently bonded, and also covalently bonded to carbons. Okay, so some additional hydrogen bonding there, you see. This is, again, notice how it is actually called electrostatic attraction here. But again, don't be surprised if you see it called a salt bridge or an ionic bond, right? So again, it's called different things that mean the same thing, which we tried to indicate when we were describing these initially. But there they are. So some of these forces that hold together, right? We're still talking about tertiary structure. Now, another thing that I would like to do, like make up an exercise. Whoops, what happened there? That I'd like to do together is to actually make up an exercise and practice this just a little bit more. Because again, this is always something I put on exams. You know, when we're talking about these types of things, is hey, or generally, I mean, I guess I can't say always, but I usually do. I usually try to simulate these exercises. So I usually try to do basically what you see on this slide. It's like, hey, let me give you a couple of amino acids, and let's just pretend that those amino acids are in close proximity, and we're still, again, talking about tertiary structure. So how would those two amino acids interact? So we could say something like this. Make sure I got the right. So let's say this. So if we had two amino acids, so let's say this. What type or types of, I guess, forces or interactions could occur between the following, I guess, sets of amino acids? Okay, so we'll look at some different. We'll throw out two amino acids and we'll say, okay, let's look at those, and I'll try to draw them. I know most of them, and if I struggle, we'll just go back and look. But we'll list two amino acids and we'll say, okay, if we put these two, excuse me, the R groups of these amino acids together, what would be the main force that would be occurring? So. let's try, let's see here. Okay, let's try these. Okay, so here's one. Glutamine and serine. Okay, so I'm gonna try to draw, and if I forget some of these R groups, sometimes I forget how many CH2s there are. I think there should be, let's see, 1, 2. I think 1, 2. Let's try this again. Okay, so there's the amino acid glutamine, and then serine is just one CH2OH. Okay, so what we're looking at, again, just to be clear, right? We're locating our R groups. Notice that these are them, right? Here are our, oh, sloppy here. There's one of our R groups, and there's the other one. And we're saying, okay, if those are in close proximity, what interaction is present? Now, I guess what we could do, just to clarify our thinking a little bit, is remember these amino acids are incorporated into a polypeptide chain. So let me draw it just a little differently. Differently here, just to kind of emphasize what I'm trying to yeah, that's okay, I guess. Emphasize what I'm trying to emphasize. So then let me try to point it in that direction. CH2, CH2, there's the carbonyl, there's the nitrogen and the hydrogens. And I guess, let me think about how that would look. That would be pointed, yeah, that's fine. Okay, point this. And let's just pretend, again, this is what I wanted to do, pretend that's part of the chain, right? It's not these isolated amino acids. I mean, that's what we drew, but these are part of chains, and the chains are coming together to be in close proximity. So if we're thinking about, now I'm going to have to kind of, let's do this. I'm going to go the opposite way for the serine. We'll do the same thing here. Nitrogen. Same thing. Okay, so we have our, and we could have pointed, I don't know why it seems like it'd be best to point the hydrogen that way. It doesn't really matter too much. Okay, now, hopefully that, drawing them like this, meaning the R groups are kind of pointed towards one another, hopefully we can see, I bet we can, right? I bet you can identify that as an H bond, okay? And again, if you want to pause the video, try these all on your own. I guess we're doing them one by one, but each time we come up with a new set of them, maybe you can pause it and give it a go, and then continue. So let's do like arginine and one of the like aspartic acid, or well, let's do glutamic acid. That's fine. Okay, so, and I don't know that I won't draw them like I drew them over there. I'll just draw the amino acids now. And if you want to draw them out kind of like we did in that last example, you can. So I always can't remember in arginine if there are 2 or 3 CH2s. Let me see. That's one of those that's like a blind spot for me. Wow. I guess I should just. So where's arginine? Oh yeah, I'm not looking on the right. There it is. 1, 2, 3 CH2s. Okay. And I wanted to use this as an example of a side chain that is often charged. Okay, and you might know where you might be able to predict where we're going with this. So there it was. It was 3 CH2s. So let me just draw this. I don't know if I'll have enough room. One, two, three. 2. 1, 2, 3. And then there was N. Sorry, that was an N. Positively charged. And then there was an NH2 there. Okay, so that's, I know it's kind of bunched up there, but there it is. There's arginine. And again, these would be incorporated into a chain. And then glutamic acid, there should be 2 CH2s. Struggling with my drawings. Whoa. And you know, we would recognize that as a carboxylate. Now, in certain pH environments, that may have an H on it, but in many cases, that glutamic acid, that side chain would be in the carboxylate form. And I'm hoping that we see, right, we have a positively charged side chain R group, right, and a negatively charged R group. So, right, this one would be, if we're thinking about the main interaction between the side chains, we would call this a salt bridge, right? Or again, an electrostatic attraction, right? If you want to think, you know, add that as a you know, a synonym here in this case, right? Means the same thing. Okay. So, all right. So let's do another. Let's continue here. Let's do aspartic acid. Do I have more room here? Oh, yikes. Do I have any more? I'll probably need more if we do, depending upon how many we do, but we'll cross that bridge. Okay, let's do aspartic acid. And glutamic acid. And calcium ion. Okay, and right away, I'm hoping, you know, based on our previous discussion, maybe right away you know what's going on, and that'd be great. If you want to draw it out, that's great too. And of course, we'll draw the amino acids at the very least. So aspartic acid just has 1 CH2. Glutamic acid has 2. We just drew glutamic acid, so we'll draw that one again. So there's the amino acid, aspartic acid. Glutamic acid just has an extra carbon in the chain. Oops. Okay, now, and plus we have a calcium ion, right? So we'll throw a Ca2 plus into the mix, and you could almost draw the dotted lines right here between, again, the O minuses and the Ca2 plus. Again, they're not, I'd rather, you know, if we did that, we would maybe draw them pointed towards one another. But in this case, right, you see the metal ion right away, and it makes you think, okay, we're talking about metal ion coordination here. For this example, okay, just to maybe delineate, right? Looking at these separately. Okay, so let's just again, I guess we've done several of them. Let's do a couple just to illustrate the others. Now, again, just as a reminder, I could, you know, I'd ask you a question just like, okay, these two amino acids, if they were, you know, pointed towards one another, I wouldn't even have to say that. I could just say, hey, here's 2 amino acids. What's the main intermolecular, or excuse me, the main force or interaction that is occurring between them? Now, what I need to do here is because I do want to illustrate the others as well. Do I have any more? Shoot. Well, let me just do this. Get rid of that. Just make a fresh one. Make sure we didn't. Okay. Yeah, let's just continue here. course, we're trying to illustrate all the 5 types. And so we could do, this is a pretty straightforward one, I think, valine and alanine. Okay, so let me just draw those for you really quickly. Valine, I always remember the what looks like the V, but it's an isopropyl group. Okay, alanine's just a methyl group, as it's R group. So I'll just draw that. So again, we've got what we're trying to recognize with this one. This would be D, I guess. What we're trying to recognize with this one is we've got 2 R groups, right? An isopropyl group and a methyl group, and they're both only made of carbon and hydrogen. So right away we're thinking, okay, those R groups are nonpolar, and so right we would say this one is hydrophobic interactions, or or London dispersion forces. Okay, we'll do one more, and if you're kind of ticking them off as we do them, you'll realize that this last one will be, well, cysteine and cysteine. Okay, this is that one that was special. I alluded to this earlier. So the cysteine is just the R group is just a CH2SH. Okay, and so if we draw 2 of those, this is the only way. Now, methionine has sulfur in it, right? So it's not the only amino acid that has sulfur in it. But remember, and we've looked at this before, it's when these two SH groups come together and oxidation occurs. Remember, oxidation can be addition of oxygen and or removal of hydrogen. And in this case, it's removal of hydrogen. What you end up with is that disulfide bond. And this is the only way, well, with amino acids, to get that disulfide bond or disulfide bridge. So if you ever see cysteine plus cysteine, right, that's a special one, and we say that one is a disulfide. Oops. Bond, or sometimes called a disulfide bridge in this context. Okay, all right. So again, and you can practice this with any 2 amino acids, right? You can take any 2 of 20 randomly and practice this, okay? And that would be a good exercise. All right, what's next here? Okay, so moving on from tertiary structure to quaternary structure, and I maybe described this a little bit earlier, but the idea here is that now we have multiple polypeptide chains, different ones, so really different proteins, as it were, coming together and being held together. So, sometimes the word that's used is they aggregate. They come together and they stick together. Okay. Now they can be pulled apart in certain situations, but in this case, they're, like for the example that you see here, hemoglobin. That's a great example of what we call a 4 subunit protein. And technically, there are 4 proteins that are linked together. And you see some. I don't care that you know that the differences between adult and fetal hemoglobin. It's just a really great example, and we'll talk more about hemoglobin in our enzymes chapter. Although it's debatable whether it's enzymatic or not, but that's not the point. The point is it's being used in this case to talk about quaternary structure. Now, I do want you to know, okay, this is important, and you actually see it highlighted in green here. These are non-covalent interactions. So of the 5 types we just discussed, 2 of them are covalent, and I want you to notice which ones. In fact, I think they're listed here. I want you to notice which ones are stabilizing influences for quaternary structure here. So these 3 are stabilizing forces for quaternary structure, because we're talking here about non-covalent interactions. If these were covalent interactions, we would be talking really about one big polypeptide chain in some ways, although they wouldn't necessarily be held together by a peptide bond. So actually disregard that last statement. But anyway, suffice it to say, we're talking about the non-covalent ones. And the ones that were covalent, remember, were the one we just looked at, the disulfide bond, okay, disulfide bridge, carbon to sulfur, sulfur to sulfur, sulfur to carbon. Those are all covalent bonds. So that one is missing here. That's not part of quaternary structure. And then remember that other weird one, metal ion coordination? I said it's also called a coordinate covalent bond sometimes. It is technically a covalent type of a bond, and so that one's missing here. So in this case, I would want you to know this, right? The three that are part of quaternary structure, holding polypeptide chains together, are H bonds, salt bridges, and the hydrophobic interactions. Those are all possible in this context. Okay. Now this is just another, again, hemoglobin is it's such an amazing molecule. It's such an integral part of really our existence. And so there's just some detail here that's good for us to know. Heme, we'll talk more about heme even in iron in our nutrition chapter, the importance of iron, getting it in our diet. Iron actually holds on to, so hemoglobin, you may know this, transports oxygen throughout our body, and it's actually the iron in this big porphyrin ring. This is just a big organic molecule, and if you go back and look at the protein, do you see the red regions here? I guess I could get my pointer. The red regions here are meant to show us that those heme groups or those porphyrin ring systems, and in the very middle of them they have the iron ion, and it's really iron that is able to latch on to oxygen, and in two different charge states, iron 2+ and iron 3+, it has a different binding affinity for oxygen, and so it's able to actually release the oxygen as well. I was trying to get rid of that. It's actually able to release the oxygen as well when it needs to tissues that need oxygen, obviously. So just some interesting stuff. Okay, now one of the last things, yeah, we're getting close to being done here, that we'll discuss is, okay, how are proteins denatured? What does that word really mean? Now, it is possible, though it's not easy. I think I mentioned this, right? This is one of my pet peeves, is trying to remove hair from a drain, right? And it's one of the things that clogs up drains. And one of the chemical ways of doing it is to actually put in some sodium hydroxide. It's in some of these cleaning agents that we dump down your drain to try to dissolve the stuff that's there. And actually a high concentration of hydroxide and some heat, and you can actually break up the primary structure, meaning separate the amino acid and disintegrate the entire protein. That, hear me closely here, that is not what we're talking about here. So we're not talking about, denaturation does not involve pulling apart the actual amino acids. It's all of the other levels, notice, right? And it's listed here, right? It's secondary. Anytime you disrupt, we've been through all of these now. Anytime you disrupt secondary, tertiary, or quaternary structure, not primary structure, you're talking about really destroying the three-dimensional conformation of the molecule, and you're talking about denaturation, okay? So make sure we're clear. It's not including pulling amino acids apart. And it's also noted here, and I would want you to know this, sometimes when this happens, it's a reversible process, meaning the protein can come together and refold properly. And sometimes when you do this, in fact, I have some examples of irreversible denaturation, and we're all familiar with a lot of these. Sometimes you just can't get these things put back together once you disrupt their structure, whatever level you're talking about, secondary, tertiary, and or quaternary. Now, you see some denaturing agents. This is just a table from our textbook. I don't expect you to memorize. Well, I actually would like you to know the denaturing agents. I should say the ones on this list. I would like you to know that they can, these things can denature proteins. I don't think I'll hold you accountable for how they affect the proteins. I suppose we could, and all of these would probably be pretty intuitive to us, but I won't make you memorize the affected regions with, for example, urea or a detergent or any of these on this list. But I would like you to know the things on this list can disrupt proteins and denature them. And again, a lot of this would be intuitive, right? An alcohol, alcohol has an OH group, so it can mess with hydrogen bonds. If you get an alcohol inside, alcohol molecules, especially a lot of them, inside your protein, and part of your protein, right? Part of these structures are, the three-dimensional structures are held together by, one of the things they're held together by is H bonding, right? Among other things. So it wouldn't be a stretch for us to spend some time exploring all of these, but I don't think we'll spend the time to do that. Okay. Yeah, and this is just so here's a good reminder. We said keratin is actually, I've talked to you about this before. Hair and fingernails and other kind of regions of our body that are structural in nature. Some of them contain keratin. Collagen's another one of these structural proteins. But remember, keratin is, or I say remember, if you look at this slide, what you'll realize is that keratin has portions of it are alpha helix. So that's secondary structure, right? And we've talked about alpha helices and how they're held together by hydrogen bonding. So again, if you have something that disrupts hydrogen bonds, what happens is, and this is one of those that's reversible, take a look, right? Water molecules, right, have OH groups in them. And so if you get your hair wet, right, those water molecules work their way into those alpha helices and disrupt them. And then as they evaporate away, those alpha helices have slid past one another. They've moved into different, well, I should say their conformation of those proteins is a little different. And when new hydrogen bonds form between side chains, between R groups, as the water molecules leave, your hair just looks a little different, right? Those molecules are, those alpha helices have slid past one another. Your hair might be now be a little bit frizzy, or it might be, curly or whatever. So anyway, just kind of a cool application here. And you see another one. You see another one here. So we're all familiar with how eggs or lots of protein-containing things like meat, right? What happens when we subject them to heat, right? Remember, if you look back at this list, heat is the top one on the list as a denaturing agent, right? And so if you heat up eggs, obviously, so the main egg protein in an egg white, and this is interesting, right? If you crack open a raw egg, that albumin, it's called albumin. That albumin is actually in solution. Okay, it's actually more soluble, and then once you heat it up, that three-dimensional conformation of the albumin actually gets kind of pulled apart irreversibly. This is not one you can put back together once you heat it up. And those proteins actually become less soluble, and they come out of solution, and you can see that they precipitate out, and that's the egg white that you see here. But what's cool about this is, I don't think I have the video linked here. I thought maybe I did. You could YouTube it if you want, but you can actually fry an egg with acid as well. And that's what you see here. It's listed down on the bottom. And if you look back to our denaturation list, or our denaturing agents list, acids are one of those things on the list. And you can actually make an egg look just like a cooked egg with acid as well. Of course, you're not going to want to eat it, but again, you can denature albumin, the main egg protein, in a number of different ways. So kind of interesting stuff. Okay, and that is the end of our proteins chapter. Now again, enzymes, which is a chapter that we have coming up, are all proteins. They're just proteins that are catalytic, or they have a function. So we'll have plenty more to say on proteins, but yeah, that's it for chapter 14.